Thank you. Today I'll be talking about how you can do more with DNA. This is the one biology slide. Cells have nuclei. We have about a trillion cells in our body, and a nucleus has 23 pairs of chromosomes. If you look at a chromosome and you look at one location called a locus, and you look at a certain type of locus that is designed for measuring variation and distinguishing people, and at that locus there'll be two different sentences. One sentence inherited from a mother, another sentence inherited from a person's father, and the systems that are used for this variation are called short tandem repeats, or STRs, and they have a word that is repeated some number of times, sort of randomly, whether it's 10 times, 15 times, and so on. And so you would inherit one repeat, say 10 repeats from one parent, 12 repeats from the other parent. If you multiply by the word length of 4, that'd be a length of DNA. And so scientists are able to measure these the content of this variation at maybe 10 or 20 different locations. And since they tend to be different between different people, it's good for distinguishing people. Okay, and then the genotype, which is the key concept here, is at that particular location on a particular chromosome, what is the pair of lengths that you have? So for example, if you have a 10 and a 12, that is your genotype replicated a trillion times throughout your body. That's one person's genotype. Okay, so that leads to a DNA identification approach where if you begin with a very simple evidence item shown on the left, then a laboratory can look at, say, 10 or 15 different locations and produce evidence data. If it's very simple evidence from one person, then you'll have one or two alleles. Either it's the same from your mother and father, or they're different. In this case, suppose that they're different. You've got inherited 10 repeats from one parent, 12 from another, and at that location on that chromosome, that's what the genotype is for this evidence. Well, when you have very simple data, it's very easy to infer over to the right what the evidence genotype is. There's only one person there. Everything in the evidence has a 10-12 genotype, and it's just easy to objectively infer what the genotype is. Once an evidence genotype has been inferred, then a comparison can be made with a known genotype, and then it can be determined if they're the same or different. If they're the same, then there's match information, where you ask at the suspect's genotype, say that's the 10-12, do we have an identification or a coincidence? Before we ever look at the data, the chance that a person has that 10-12 genotype at a locus is, say 5%. That's in the denominator on the right. That's the probability of a coincidental match. After you look at the data and you see that the evidence has only a 10-12 there, and there's a match between the evidence and the suspect, there's a 100% chance of a match. That change in information going from before to after is what the data does, and then you get a ratio, which has many names, including likelihood ratio. And by going from 5% in the denominator up to 100%, that ratio of 100 over 5 is 20. That's the match statistic, or the LR, likelihood ratio, at one locus. And you can imagine if you multiply 10 or 15 numbers like 10 times each other, you get very big numbers. That's what match statistics are like. However, most DNA data these days is not simple single source data that comes from one person where it's obvious what the peaks are trying to tell you. Instead, you can have degraded DNA, low quantities of DNA, or very commonly mixtures of 2, 3, 4, 5 people contained in the same evidence item. And this is what DNA data looks like. What you're seeing here are 4 peaks. From left to right on the x-axis is the size, and as the number of repeats, which you see in the boxes on the top of each peak, increase, with a 7, 10, 12, and 14, that's the number of repeats. It's the allele, and there are 4 of them, meaning there are at least 2 people. And the height of the peak on the y-axis indicates the quantity. So at a peak, that comes off the DNA sequencer tells you is which allele it is, say a 10, and the relative quantity of DNA. What happens, though, is that because this now has to be interpreted, there's no longer one possible genotype out of locus out of 100 possible pairings of, say, 5 and 6, 5, 7, 5 and 7. About 15 alleles, there'll be about 100 pairings. What you end up with instead is data, and people generally do not infer the evidence genotype. So that pathway is no longer there. Instead, approximate statements are made about the data peaks themselves. But if you don't have an evidence genotype, as you see on the right, then you can't make an exact comparison with a genotype. And so this DNA pathway for evidence and DNA databases breaks down. What are the issues with human interpretation? I'm sure we'll hear a bit more from Alleles, I hope, and possible bias and issues. But here are the key issues with evidence. The first issue is that much excellent DNA evidence data is called inconclusive. In the States, you might have maybe half of the complex mixtures just called inconclusive. The data would be informative if you can interpret them, but people by eye can't. And so the evidence is just not used. The reasons are that the peak heights that they're looking at might be too low for them to analyze within the interpretation guidelines they have. There are also maybe too many contributors there. Beyond 2 contributors, it gets a bit wonkier, and there's a tendency to apply a guideline in a laboratory that says we won't give any sort of match statistic. We can't go much beyond what we're comfortable doing with taller peaks and only 2 contributors. There's also a potential for examination bias if a person is looking at a result, because if you have the data in front of you and you're looking at a bunch of peaks and you're comparing it with what alleles would be from a suspect, you might be more inclined to see something in the data. If you go all the way and infer a genotype, write it down, and do that without knowing the suspect genotype, that would be more objective. It also has implications for databases. The UK National Database, as well as POTUS in the US, find their hits between evidence, lists of alleles, and potential criminals just by an association. There's no match statistic computed. So the information content isn't computed when the match is made. You may get 10 or 100 names back, therefore. And the result of that is you have more names in the comparison, you can get false hits. If there's only one person who contributed to the DNA and you have 100 genotypes coming back, then clearly at least 99 of them are not right. And the solution to that, certainly in the US, is to restrict upload so that only more informative mixtures can be used as evidence. That will lead to maybe only 20% of evidence samples even making it up to a DNA database to potentially make a true hit. TrueAllele is a computer system that has a rich statistical model that's been developed for about the last 15 years. And what its goal is with evidence is to preserve all of the data information. It uses all the peaks, whether they're high or low. It doesn't touch the data. It just infers from data. It can work with any number of contributors. So if you have the world's population in the evidence, is not going to help very much. But typically for 3, 4, 5 contributors, the models tend to work well. And the method is entirely objective in the sense that the system does not know, nor cannot know when it infers a genotype, anything about who you're comparing it with afterwards. So there's no potential for examination bias. There's a corresponding database system that's built on the same principles, and hits are based on likelihood ratio match statistics. It's very sensitive that it finds true hits, and it's very specific. It's only true hits without false positives. And the result of this is that the DNA information pathway is restored, because regardless of what that data is, no matter how weird it looks or how many contributors may be in there, the machine can always infer an evidence genotype. Now, there's uncertainty. With single source data, you started with maybe 100 possible pairings of alleles and ended up with only 1. That's what perfect data will do for you. With this sort of mixture data, you will reduce that list maybe to 5 or 10 possibilities instead of 100, and there'll be information. But to represent that uncertainty, like in the rest of science, we use probability, and the computer may spend half a day or a day on an interesting problem working out what those probabilities are for every contributor at every genetic locus. But once you have an evidence genotype, which in this case is known up to probability, a comparison can get made to a known genotype. And the result is that using likelihood ratios, you can preserve the match information. And the reason why is that the data is still updating your information about to what extent there's an identification. The coincidence stays at 5% at the suspect's genotype, but now the probability of a match may have reduced to only 50%. That's not a problem for likelihood ratios. Instead of dividing 100 by 5 to get a match statistic of 20, you divide 50%, say, by 5, and you get a lower match statistic here of 10, but it's scientifically valid. So I'd like to tell you about one of the cases that we've worked on. This was presented at the ProMega meeting, which is an annual DNA meeting in Nashville, Tennessee this fall, and it's about a gang crime in Kern County. And what happened is that Kern County is located in southwestern California, about 2 hours northeast of Los Angeles. So it's on a corridor between Northern California and Mexico, and they have an issue with drugs and other crimes and criminals going north-south, and people stopping off for lunch, hitting up a food mart while they're there. And so they have a lot of interesting criminals who wander through. And some people stay. I guess that's a form of immigration. And they create gangs. This was one particular gang, and they began slowly. There was a food market, and they robbed the store, and they left behind some items. This was a gun and a hat, from which some biological evidence was recovered. And then they continued moving through the county. They went to a hardware store, and they left DNA on a safe. They left behind a mobile phone. And they didn't stop. And as they escalated, the crimes became more violent, more abusive to the victims. There was a jewelry store, which we'll see a video of in a few minutes, where DNA was left on a countertop and on a safe again. And this gang just became more difficult as time went on, and it became a goal of the police and prosecutors to not only stop the gang, but convict them of enough crimes so that in California they would stay in prison for a long time. If you're only convicted of a crime that has 5 or 10 years as a sentence, since there's overcrowding, you may be released quickly. So there's all these items of evidence at these multiple scenes, and as they're looking at the DNA, this is the DNA from one item, and this isn't data, this is what's actually in the DNA. So this is what genotypes look like if you draw a picture of them, where the x-axis is the size, it's the allele, 13, 14, 15, 16, 17, and so on, the number of repeats. The y-axis is the relative quantity. So what's actually in this evidence might be shown in blue, a first contributor who's the major contributor with a genotype of 13, 14. A second contributor shown in green who's a minor contributor with a genotype of 18 and 16. And then a third contributor who's also in the DNA with a genotype shown in orange of 17 and 20. So this is, suppose, the true state of nature that we're going to try to recover by doing experiments to generate data and then infer from. So what the lab does is it generates STR data, short tandem repeat data, in this case for about 15 chromosome locations on each item of evidence. And you can see that the data is indicating something that looks a lot like the pattern, right? You have tall peaks on the left at 13 and 14 circled in blue, and you have medium-sized peaks shown in green, lower peaks shown in orange. So that would be a good hypothesis as to what the genotypes would be from the 3 different individuals at this location, genetic location, but it's not science yet. It's just a suggestion. It's a hypothesis. A lot more work has to be done to interpret it. This is what the laboratory processed. They had 12 evidence items from 5 different crime scenes. We had gone through the locations of those before. Again, the hack, the gun, the safe, the phone, and so on. So they did the STR work in the laboratory, and the data that they generated were almost entirely 3- and 4-person mixtures that had very low quantities of DNA. So the Kern County lab decided that they would not try to even interpret this by eye, but they would bring in a computer for this. There are also 10 evidence items, 10 reference items shown on the right, 5 from victims and 5 from suspects. And the task was to make a comparison between all 12 evidence items against all 10. Just wait for it to finish, if you don't mind. Okay, so the question is, if we have 12 evidence items, and we have shown here are just the suspects, 10 reference items, there are 120 comparisons that would have to be made in order to determine to what extent an individual is included or excluded, as reflected by the likelihood ratio, which is the match statistic. And again, that was a lot of work. They didn't feel they would get anything by doing those 120 comparisons. Would just tie up the lab's personnel for a few months. So human review doesn't produce results in these sorts of cases for several reasons. One of them is that people apply thresholds to the data. Instead of looking at patterns where you can see what's tall, what's medium, what's low, and how the data arranges itself, a line is drawn at some level that the lab sets based on calibration studies. And everything above that line is considered to be have equal standing. So the peak heights aren't used. It's are they present or absent. And any of the data that's below the line is used less or ignored, depending on the method. So if it turns out that there's a minor contributor with a genotype of 1618, shown with the green arrows, that data would not be available for inferring the genotype. So people do this because these are very hard problems. If you were to consider that there might be 100 possible allele pair genotype values for the first contributor, 100 for the second contributor, 100 for the third contributor, there are a million possibilities of what the genotypes can be, and they arrange themselves based on different peak heights, different quantities of DNA. There's a lot to consider. And if you replicate this for 10 or 15 different loci, you can't expect people to work on problems that have a trillion different answers. And so they make approximations. And they work very well for simpler mixtures. But this is how the computer thinks. TrueAllele is a fully Bayesian model with several hundred variables. And I'm only going to focus right here at one locus on the genotype variable and the relative quantities of DNA. There are other variables for stutter, PCR artifacts, variation of all parameters, and so on. So what the computer does, as you can imagine, is that it tries out by statistical simulation, Markov chain Monte Carlo. It's a computer method that is used for tens of thousands of problems. It's been around for maybe 50 years. It tries out almost every explanation. What you're seeing here is a very good explanation in that the rectangles shown in color very much correspond to the peaks that you see underneath. Do you see that? It's the pattern. If the computer were to propose that there's a lot of the first contributor, 13, 14, and put some blue rectangles there, that's just for us to see it. A little bit less of the second contributor in green, and a lot less of the third contributor in orange, at just those locations and those amounts, then the picture that you're seeing looks very much like the data that's underneath it. So that sort of explanation would have a high likelihood of being true, because the pattern that the computer is proposing of those 3 genotypes in those amounts explain the data. It produces a pattern that looks like the data pattern. But the computer is also proposing tens of thousands of other patterns that don't look like this. It can give different quantities. the 13 and 14 might be a tenth the height. It might be that there's nothing on 13 and 14, but instead the computer is proposing an 11 and a 12 here and here, and putting nothing where the data are. If it's not explaining the data, then there's a lower likelihood, probably is virtually zero in this case. And so by assessing, by simulation, hundreds of thousands of possibilities, the computer gets a sense of which genotypes and their combinations produce patterns that better explain data. Those genotypes will end up with a higher probability. And those patterns and genotypes that don't explain the data, they're creating patterns where the data aren't, and those will end up with much lower probability. So after the computer thinks about this, looking at all the data for a day or so, it will produce, without having looked at a suspect or anything that it would compare with, objectively inferred genotypes that are not biased by knowing an answer for each of the three contributors at every locus. So here, for one of the contributors, is what a genotype looks like that was inferred in this case. There's an allele. Here's the listing of allele pairs: 16, 18, 14, 18, 13, 18, 18, 20, 17, 18. Of roughly 100 possibilities, these comprise about 99% of the probability of just these 5 allele pairs, and there are probabilities associated with them. Once you have a genotype that has allele pairs with the probabilities, then match statistics can be calculated. And that's what the computer does. Virtually all the computing time is in getting the genotypes to do a comparison between the 12 evidence genotypes and the 5 suspects. While it might take a person working visually many weeks to make a comparison, the computer can take the genotype of say, suspect 1 relative to the genotypes of the gun, do a comparison relative to a random population, and see that there's no statistical support for a match. In this case, we see that there are 7 positive matches out of the 60 comparisons that were made. Again, this saves people from a lot of frustration because most of the time you're not going to get anything. Some suspects appear in multiple items of evidence, like in the last column. Sometimes one item of evidence, like the hat in the second column, it corresponds to 2 different suspects. What the number is, it's the number of zeros in the match statistic. It's easier to write down the number 3 than it is to write out 1,000. It's easier to write down the number 6, shown the counter relative to the second suspects, than it is to write 1,000,000. So scientists tend to represent information by the number of zeros after the 1 in the very big number, also known as the logarithm of the match statistic. So that's one way of looking at the answers in a table. Another way is to draw a picture, which we did for this, certainly for this conference presentation. And what you can see are lines that are connecting the different crime scenes with the different people. You see that, for example, that the hat at the food market is a mixture of suspect 1 and suspect 5, which gives you more probative information that these 2 individuals were located at the crime scene. You also you get a sense that individual number suspect 5 was more actively involved, or maybe he's a DNA shedder, but the theory of this particular gang was that he was much more hands-on and did the work. You also see a match to a counter in a jewelry store, and that's what led to a result in this case, which not there yet, which is what I testified about in January. And there are many ways to describe a likelihood ratio, and they all have different English phrases corresponding to different formulas. The formulas all give you the same number, but this is a way of phrasing a match statistic that juries, certainly in America, find understandable. And it goes like this: a match between the evidence and the suspect is 500 million times more probable than a coincidental match. That sounds like English, and the words actually correspond directly to the math. Words like match, coincidence, and so on. probability. And this is described in an article in Forensic Magazine. This talk and the slides will be on our website, which I'll have the address for at the end, so you'll be able to see that. So I'd like to now show you a news clip that was made out in Kern County, I guess in February after the trial.
New at 5:30, prosecutors are hailing new DNA technology that helped secure 2 convictions in a string of recent robberies.
The new TrueAllele casework system made its debut in a Kern County courtroom this month. 17's Kiyoshi Tomono shows you how it works.
Charles Lawton and Dupree Langston might have gotten away with robbing this check advance store if they hadn't done this, planting a bare hand on this countertop to vault over. In the fraction of a second Langston's sweaty palm touched the counter, it transferred a minute amount of DNA that would lead to his conviction.
And so then the technicians went back to that very spot and did some swabs.
Before, the crime lab might have struggled to do anything with evidence like this. Only a trace amount of DNA and contamination from other people who touched the same counter.
And that used to be the biggest problem, was when you had a mixture of profiles. And so you couldn't pull out exactly who was in there, who wasn't in there.
So in those cases, we would have to say that the DNA profiles were inconclusive in our analysis. With TrueAllele, we don't have to say that any longer.
TrueAllele is high-powered computer software that uses mathematical formulas to sort through contaminated evidence.
This is the first use of TrueAllele west of the Mississippi, really.
It can then pinpoint with a high level of confidence whether a particular person's DNA was present at the scene of a crime, even if they barely brushed an item.
We're talking about touch DNA, we're talking about DNA with that's been out in the field for a while and may be degraded. We're talking about missing persons cases, potentially.
It is a powerful tool with a caveat.
DNA, it is time-consuming and it is expensive. And so we can't do it for every case that we would like to.
In all, Lawton and Langston were convicted of 8 robberies in a 5-week crime spree, including a holdup at this Max Muscle store in 2011, where the owner shot back. Other victims said they were brutalized. Some were even forced to disrobe at gunpoint. Now that the technology has been proven in court, the crime lab says expect to see more of it.
With newer technologies, we're able to give more definitive answers.
In downtown Bakersfield, Kiyoshi Tomono, 17 News.
And a footnote here, Langston and Lawton are set to be sentenced on April 5th. They each face about 70 years in state prison. The new DNA software, by the way, was paid for with a $100,000 grant from the National Institute of Justice.
So to establish that the system works as it's supposed to, Cybernetics, in collaboration with crime labs and crime labs not in collaboration with Cybernetics, do a lot of validation studies. There have probably been at least a dozen studies done, and more are continuing. Every time another lab brings TrueAllele on board, they like to do another 1 or 2 studies. Here are 3 such studies. They usually assess the sensitivity of the method, that is, what is the match statistic that the computer is getting? Is it able to demonstrate that somebody who's believed to be in the evidence is included? They also assess the specificity, which is the degree to which the system doesn't get the wrong person. That is, you don't want it to get match statistics that improperly include someone. And they also assess the reproducibility. I'm not going to go into all these studies, but I will describe one picture from the most recent one that's in press that explains quantitatively how TrueAllele gets the right answer, doesn't get the wrong answer, and when the computer repeats the question is reproducible. So this is part of a study we did with New York State Police. They're currently bringing out their TrueAllele system for use in Albany that covers about 8 million people in New York. And let me describe to you what you're seeing. On the x-axis, we've just listed the 81 evidence items and the roughly 85 genotypes that were inferred that matched something in the study in descending order, shown by that blue cloud in the background. That blue cloud is what the computer did, is what TrueAllele did. The vertical axis is the log of the likelihood ratio, the number of zeros in the match statistic. So for example, 15 is a 1 followed by 15 zeros, or a quadrillion. So, and if you just look at the blue cloud in the background, that was the information, identification information obtained by the computer. The average match statistic, if you look at the center, halfway across, just draw it around there, is about a match statistic of 10 to the 15th, 1 followed by 15 zeros, or a quadrillion. In this study, a comparison was made with human review on the same evidence items, and using 3 different methods depending on which protocol the laboratory followed. In gray, the computer got the same answer as the quantity of information as a person. This is a method called RMP, where a person under certain conditions will look at a mixture and say that there's enough of the major contributor, maybe 90%, that they can ignore the minor component and treat it like it was just easy single source data. In that case, the statistics agree. In about 8 of the cases, shown in green, there's a method that's actually similar or identical to what's done in the UK, which is the combined likelihood ratio. It uses thresholds. It's not using quantitative peak information, but it uses more of the information. For example, like that the suspect is present in the evidence. And what you see in those cases is that it gets about 2/3 of the information. In orange, there are about 12 scores, or 12 human results, that give about 1/3 of the match information. That's using RMNE, random man not included, that just looks at the different peaks as if they were alleles. But what's most striking about this is that people did not get a result in 70% of the evidence items. So that's how much of the information has been discarded. So 30% of the cases, they could make a statistic that would bring an item into court, and the remainder, the evidence would have gone unused. TrueAllele has been used in criminal trials. I've written over 100 case reports that have been filed on DNA evidence and given testimony in U.S., state, federal, military courts, as well as overseas, like here, for crimes of a full spectrum: armed robbery, child abduction, murder, rape, terrorism, weapons, and so on. TrueAllele is used in 2 ways in the United States. In blue, these are states where 1 or more labs have purchased a TrueAllele system, and they're bringing them on board to use in-house. And in those states, as well as in the orange states, groups have cybergenetics interpret the evidence for them because their system isn't on board yet, or they don't have a system. So that's TrueAllele in the US. In the United Kingdom, cybergenetics was first here in the year 2000, and we did work for 5 years with the Forensic Science Service, automating the DNA data bank, and deployed the first fully automated DNA expert system for reference samples. It was faster in that it eliminated the 350,000 item backlog. It was better in that the human error rate of the FSS of 1 in 2,000 samples went to undetectable. And it was cheaper in the sense that 2 desktop computers at that time, for the simpler problem, eliminated about 100 jobs at Priory House in Birmingham. More recently, in 2010, we've been working with the Police Service of Northern Ireland with TrueAllele Casework, which is the system I've been describing, producing match statistics for very complex DNA evidence, typically with 3 or 4 person mixtures on low template and degraded DNA. We've done about 10 cases with them, including the Mazarine Barracks attack, which I testified at in Belfast a little over a year ago. The concept of preserving information really works even better for investigative use with DNA databases. Currently, most mixtures in the US don't make it onto investigative DNA databases, and virtually no mixtures make it up in the UK because the system's not designed for mixtures. But if you could interpret all the mixtures from crime scenes and build a genotype database, take the amazing information on your national DNA database and upload that as genotypes, and had automated matching and reporting, you'd be making much better use of the data you already have for making cold hits and apprehending criminals before they commit new crimes, as well as solving old ones. So in this TrueAllele computer age, we have an objective and, by science, reliable truth-seeking tool. which solves DNA mixtures as easily as people solve single source problems. It handles low copy and degraded DNA, and it provides accurate DNA match statistics, automating the interpretation. And it's currently used for basically everything you'd use DNA for. Learning backlogs, reducing costs by using machines instead of people to solve problems that are more appropriate for machines, solving crimes, finding criminals, convicting the guilty. We also work on defense cases of actual innocence and creating a safer society. If you're interested in more information, we have courses on our website, newsletters, recent cases, presentations like this one are put up as slides, handouts, and often as narrated movies, as well as all of our publications that we put out. And if you're interested in seeing how the system works on data where you have maybe a 3-person mixture, it's an important case, you can see the data, or your DNA analyst in the lab can see the data, but they can't put a number to it, then you can send that to us and we'll take a look and let you know, just for fun, what it is that might be there, and use that as a starting point to tell you more on cases and data that you care about, what the system does. Thank you.